All right, still see some attendees joining, but we'll go ahead and get started here since we have a lot to cover today. So I'm Tess. I am founder of Renegade Research. We're a decentralized patient and caregiver-led nonprofit research organization. We conduct distributed research and educational projects like this seminar, and we're building a network designed to improve access at all levels of research and care. Our largest project is Remission Biome, and we're also organizing other projects, including topics like heart rate variability, anesthesia, postprandial symptoms, and more. Our educational projects include research roundtables like this, and also clinical roundtables. You can find out more at remissionbiome.org. You can subscribe to our newsletter. And if you would like to donate towards Renegade Research, we do have a link on our website. I'll also put my email in the chat if you want to reach out. We have a large team of over 30 people, and many are here today as panelists. So thank you all for making this possible. So our speakers today are working together on a research projects awarded by the Patient-Led Research Collaborative. The research fund is a project of PLRC, the Research Collaborative, and funded by Balvi. HiFiBiO is utilizing its single-cell profiling capabilities to analyze samples from long COVID and ME/CFS patients. Together with the Selin lab, their goal is to acquire deep insights into underlying immune mechanisms of these illnesses and understand the diversity in the patient population that could lead to potential diagnostic biomarkers and therapeutic innovations. Additionally, today, the Selin lab will discuss their new paper, Identification of CD8 T cell dysfunction associated with symptoms in ME/CFS and long COVID, and treatment with a nebulized antioxidant antipathogen agent in a retrospective case series. HiFiBiO Therapeutics is a clinical stage global biotech pioneer, pioneering a unique high-resolution translational platform to enhance the probability of success for immunotherapies. The Selin Lab is a viral immunology lab at UMass Chan Medical School focused on understanding human adaptive immune response. Liisa Selin is an MD, PhD, and her colleague Anna Gil, PhD, studied T cell response in multiple neuroinflammatory infection-associated chronic conditions and illnesses, such as ME/CFS and long COVID and MS. The lab seeks to conduct clinically significant research in order to advance diagnostics and treatment for these complex diseases. Liisa is a professor in the Department of Pathology and is a fully trained infectious disease specialist who has conducted more than 30 years of research focused on these chronic diseases. Specifically, she has published many articles on the role of T cells in response to viral infections. Specifically, she conducted research that initially identified and focused on the phenomenon of heterologous immunity, whereby memory T cell responses to pathogens can influence the outcome to subsequent infection with unrelated pathogens. She has ME/CFS. Anna Gil is an instructor in the Department of Pathology, also at UMass, and has worked closely with Dr. Selin for the past 14 years. Her focus is on viral immunopathogenesis, and she has done seminal work in the role of T cell receptor repertoire in disease outcome during viral infections. More recently, she has focused on the role of T cells in the immunopathogenesis of chronic conditions such as MS, ME/CFS, and long COVID. Roshan Kumar, PhD, is an executive director, the head of external innovation at HiFiBiO Therapeutics in Cambridge, where he works to identify novel drug targets for the company's pipeline of immunomodulatory therapies and develop collaborations with academia and industry. Roshan's interest in ME/CFS and related illnesses began when his wife fell sick 20 years ago. He and his team were recently awarded a grant from the Patient-Led Research Collaborative to work with the Selin lab at UMass to profile adaptive immune repertoires in patients with ME/CFS and long COVID. This work will help define immune dysfunction that may contribute to the development of disease, leading to new biomarkers and therapeutic targets. Megan Fitzgerald is a PhD in neuroscience and neurology. Megan became sick with long COVID in 2020 and now focuses her research on that condition. She is the lead of the PLRC reinfection study and a patient rep for 2 PLRC-funded research projects, the Selin Lab's T cell research and also the HiFiBiO Selin Lab collaboration. Rivka Solomon is an ME and long COVID patient advocate, community organizer, and writer. She also works with members of Congress, the media, doctors, researchers, and a number of organizations focused on infection-associated chronic illness. She is a patient rep for the Selin Lab's T-cell research, as well as for the HiFiBiO Selin Lab collaboration on single-cell immune profiling. Rivka has had ME for over 3 decades. So I'm very happy to introduce everybody, and I'm really looking forward to these presentations today. I will go ahead and pass it off to Liisa, if you want to share your slides now.
Sorry, I was muted. Thank you very much for that very nice introduction. And I'm going to begin this little meeting presenting our work that we just recently published on CD8 T cell dysfunction in ME/CFS and long COVID. This came out in Brain Behavior and Immunity this past year. Our hypothesis is that ME/CFS and long COVID result from an aberrant response to an immunological trigger like infection, leading to a dysregulated immune system with overactivation of CD8 T cells and subsequent exhaustion of these T cells. We will also test, as I will show you data on whether act as an immunomodulator to reverse the state of T cell exhaustion and help improve symptoms in a retrospective case series. This is not a clinical trial. First, when we set up our biomarker studies for ME/CFS to study immunopathogenesis, we had to at that time show that we think ME/CFS and long COVID are basically the same disease or very similar diseases. And this was borne out using a new questionnaire that we designed for our lab because being a patient, I wanted something that was a little bit more simplified and took a little less energy to fill out. And as you can see here in the gray bars, the striped gray bars are ME/CFS and the black bars are long COVID. The symptom profile for all the symptoms are very similar, such as the common symptoms of fatigue, post-exertional pain, postural hypotension, difficulty concentrating, and the overall severity score by the patient was very similar. There were some differences in things like cough and chills, as one might expect with long COVID. And all of these symptoms were significantly different than healthy controls, which are these tiny little gray bars at the bottom. We then went on in this patient population, a subset of them, to look at their function of their T cells and using an intracellular cytokine assay stimulating with PNA and iomycin. And on this graph, you can see interferon gamma production here on the left, TNF on the right. And you can see that the long COVIDs in black and the ME/CFS in the striped bars are significantly lower than the healthy donor controls in their interferon gamma production. Okay, sorry. Transforms. So you can see here is just a review picture from a review article showing that immunological and metabolic pathways have been shown to be, by many others as well as us, to be highly dysregulated in long COVID. And among the papers published recently in this area are one by Akiko Awasaki, and even the large study that came out of NIH recently had a section showing that there was evidence of CD8 T cell exhaustion in both of these papers in Now my key's not working. Okay. And there have been other papers showing long COVID T cell dysregulation from the Rowan group. And of course, Nancy, thank you. So there was a study by Maureen Hansen showing that there was a form of oxidative stress going on in the T cells of ME/CFS patients that was highly consistent with CD8 T cell exhaustion. She has subsequently also done studies to show T cell exhaustion during ME/CFS. So we then went on to do a collaborative study with patients. It's not even a collaborative study. We had access to doing a retrospective study looking at this new treatment in Fitzgerald, which is a multi-mechanism combination drug that reestablishes antioxidant-oxidant homeostasis, modulates immune responses, and eliminates inflammation with broad-spectrum antibacterial and antiviral properties. And it contains, it's a liquid formulation. It's taken by nebulizer, and it is comprised of 5 strong antioxidants that you may have heard of before, such as glutathione, N-acetylcysteine, methylcobalamin, and newer compounds like 1,8-cineol and beta-caryophyllene. And the one big advantage of this treatment is that because they're taken by nebulizer, you can take very tiny quantities. They get very high systemic levels. They get very quickly to the brain and have very few side effects because of this. So in this retrospective case series, we looked at 4 ME/CFS patients and 4 long COVID patients. We found there were no significant side effects other than mild cough that could be controlled, and all their routine blood work remained normal. And these patients were all patients of Dr. Salerno, and the drug was made by his compounding pharmacy. Here is a slide that shows the symptom profile of one of the ME/CFS donors. This donor had been actually relatively mild until her first dose of Pfizer vaccine, and she became extremely ill with neurological symptoms and extreme fatigue, difficulty concentration, couldn't work, and on bed rest. And immediately upon taking Inspiritol, her symptoms started to very rapidly improve. And over 3 or 4 months, she was 50% improved, and over the 15 months has gradually improved, although she did have, following the vaccine and the T cell exhaustion, aggravation of it, she did have evidence of chronic shingles and chronic cold sores, and had to also have therapy with antivirals. Here is one of the long COVID, one of the more severe long COVID patients who had been a 62-year-old female, had been totally healthy until she got acute COVID, and then was highly symptomatic with the common symptoms of fatigue, headaches, difficulty concentrating. She took her vaccine, and a Pfizer vaccine, got first, and then went on this Inspirital therapy, and actually probably improved a little bit faster even than the ME/CFS patient. She took the second vaccine because of employment reasons, although it was recommended she shouldn't, and her symptoms were exacerbated. She was found to have IgM positive EBV and CMV, and had to go on vanganciclovir as well as Inspiritol. And once again, her symptoms have improved. We then looked at the interferon gamma and TNF alpha production by both these patients and all the other patients in the study, and found that they had significant improvement in their interferon gamma production. This is pre-Inspiritol, and this is post-Inspiritol. And they had about a 3-fold increase in interferon gamma production, and we had similar results for TNF. When we looked at the symptoms of these 8 patients, And here you can see, as on the previous slide, the red is ME/CFS and the blue is long COVID. We took their overall symptom severity score, and there was a 50% reduction in the severity of the symptoms of these patients. And here's an example of one of the specific symptoms common in these diseases, post-exertional malaise. There was also a significant decrease in that. We've done some studies recently to add Inspiritol directly into the intracellular assay. These are still early works, but we are seeing enhancement of interferon gamma production directly, suggesting Inspiritol is acting directly on the CD8 T cells to help reverse exhaustion, and we're very interested in pursuing this. So the clinical implications of these studies for diagnosis and treatment of ME/CFS and long COVID. We've shown evidence of a potential biological marker for diagnosing and tracking a response to therapy, both for ME/CFS and long COVID, characterized by functionally exhausted CD8 T cells with severe deficiencies in their ability to produce interferon gamma and TNF alpha. The Inspiritol therapy, which is an immune modulator containing 5 antioxidant antipathogen compounds, may be useful taken by nebulizer, goes directly to the brain from the lungs, scavenges oxygen radicals, is known to attenuate NF-kappa B signaling, and that is one of the ways we think it's helping reverse exhaustion. It has antiviral activity, and therefore altogether, it helps with reversing exhaustion and improve symptoms. The company is presently awaiting FDA approval and is actively trying to set up clinical trials. Inspiritol improves CD8 T cell function in vitro. And we also wonder, due to CD8 T cell exhaustion, ME/CFS patients, do they have difficulty in controlling persistent viruses, particularly the herpes virus family, such as EBV, HSV6, CMV, and HSV1? And thus, would antivirals or antibiotics, antifungal, or microbiome therapy be helpful? And then there's always the question of checkpoint inhibitors for CD8 T cell function, but which have been useful in cancer, but I think we still have a long way to go and need to be very careful since the exhaustion is systemic rather than in the tumor. Our future directions is to identify other new biomarkers, and we're actively working on this, which may stratify patients based on aberrant immunological compensations they make in response to CD8 T cell exhaustion. We are finding at least 50% or more of the patients have increased CD4, CD8 cells. There are some patients that have increased MATE cells, and others have increased gamma delta T cells, and a portion of the patients have increased cytotoxic CD4 T cells, and we're very interested in stratifying these to severity of disease. We're also beginning to examine NK cells and their functionality as part of our biomarker, and hopefully I'll be able to update you on those in the future. But in collaboration with Roshan Kumar, who you're going to hear shortly from HiFiBiO, we will combine our expertise to examine in depth in the same patient population using single-cell RNA-seq, identifying genes of interest and tracking TCR repertoire in order to help identify potential persistent pathogens like EBV, CMV, VZV, or HSV, which may be unique to each patient. So you're going to hear all about that from him next. And these are, of course, all my collaborators. Anna Gil, who's completely essential to everything that we're doing here. It wouldn't be possible to do any of it without her wonderful work and expertise. And joining us in the past year has been Nura Aslan, a postdoctoral fellow who'd worked with me previously, who's very dedicated also to understanding ME/CFS. She came all the way from Turkey to join us, and our technicians, Teva, Rivka, and Megan, who were introduced here and you'll hear from, are absolutely wonderful and essential to the progress of our research. Cynthia Adnick helps us also as a patient advisor, and the ME Association wouldn't be possible. They helped us get all this geared up really fast. And then our other collaborators, including Nancy Klimas, Mady Hornig, Inspiritol, and long list of them. And of course, Roshan Kumar. And all of this work has been funded, given by the Ramsey Award is how we first started. Then we got our R01, and now the patient-led initiative and private donations have kept us going when things have been tough. And I have a slide, if anybody's interested, on my model of exactly what I think is going on in ME/CFS and long COVID. I can share with you during question period if anyone's interested. So thank you.
Thank you. And I think we'll definitely be interested in seeing that during the Q&A. So next we'll have Roshan. And I'll also mention, we'll go through all the questions at the end. So Roshan, if you want to get your slides up, and I'll just mention that if you click the Q&A button on the bottom, you can type in your questions, and you can upvote questions so that we can prioritize which ones to ask at the end.
Great. Thanks, Tess, and thanks, Liisa. Let's see, can you all see this full screen?
We can.
Yeah.
Okay, terrific. And thank you to Remission Biome for having us here today and giving us an opportunity to share our work with you. It's really nice to be able to participate in a forum like this. I'll start out by telling you a little bit about our company, HiFiBiO Therapeutics, and how we approach immune disease biology and why we're interested in this project. So we are an immunotherapy company. We develop therapeutic antibodies. We're based in Cambridge, Massachusetts. You can see our vision, mission, and strategy here. We look at biology through a single cell lens. So we have our own platform for looking at individual cells. We call it DIS, or Drug Intelligence Science. And we really believe by understanding the heterogeneity of individual cells in different diseases, that we can understand mechanisms, we can identify particular disease pathways operating in different patients, and really tailor treatments to treat their particular disease state. And so really our goal is to develop a sustainable pipeline of innovative immunotherapies using this approach. We have identified to date 8 immunotherapy candidates. These are both for immuno-oncology, for treatment of cancer, and for autoimmune diseases. We've advanced 3 programs into phase 1 clinical trials for the treatment of cancer. So these are immuno-oncology programs with antibodies targeting immune checkpoints or stimulating immune cells, such as T cells, to activate an anti-tumor response. We have 2 IND facing, so getting ready for progressing into the clinic, immunology and inflammation programs. So these are for the treatment of autoimmune disease, and one of these INDs is anticipated later this year. And we've had a number of partnerships, including out-licensing some of these drugs to other companies. And we are fortunate to be recognized as one of the Fierce Biotech Fierce 15 last year. So really our goal is to develop innovative immunotherapies. And part of how we do this is using this platform that we call DIS, Drug Intelligence Science. So what this is really, it's a suite of tools and platforms to look at single cells and understand their role in disease biology. So this includes some hardware. We have a droplet-based microfluidic system for analyzing single cells that we've developed. So you can encapsulate individual cells in these drops and then sequence them to look at the state of each cell, as I'll tell you about. And then we've spent a lot of work with building up our computational team, integrating single cell data to build up disease atlases in cancer and autoimmune disease, and then developing and applying computational algorithms to mine and explore these data. So really what we try and use these platforms for is to go from patient samples to identifying new targets, novel drug candidates that can advance to the clinic and be new medicines, and predictive biomarkers for patient stratification. how do we become interested in ME/CFS, and why do we think applying this sort of approach to ME/CFS and long COVID can be fruitful? As mentioned in the intro, I had a personal interest in ME/CFS through my wife's experience, as she had been sick for many years, and I gained an understanding of sort of the lack of insight that we get in the clinic to the immune system and what's actually going on in patients who can be very sick, but many of their standard clinical tests look normal. And you just heard from Liisa, clear functional tests that her lab has been doing that show clear evidence of T cell dysfunction and abnormalities in ME/CFS and long COVID. And with some of the references she cited and other work being done in the community, you can see there's more and more evidence of T cell exhaustion and dysfunction in these diseases. So what we are doing is applying this single cell approach to profile immune cells from patients with ME/CFS and long COVID. And we are applying single cell sequencing to these cells. And what that lets us do is it lets us profile their functional, their state, and because we catalog the genes that they are expressing. And each cell type is going to have a somewhat unique gene expression profile that is related to the function and state of those cells. So exhausted cells are going to look a particular way, and it's different from other T cells in other states. And at the same time, we are profiling the immune repertoires of these cells. And what does that mean? Well, it means their B cell receptors, which are their antibody sequences for B cells, and the T cell receptors for T cells, which is how they recognize antigens. And the antigens are the parts of foreign pathogens that these T cells recognize. And what we're very interested in doing is using these T cell receptors to get a handle on the antigens recognized by these cells and use that to gain insight into disease biology. So when we talk about functional exhaustion with T cells, typically it's because they're responding to an antigen that is continuously stimulating those T cells. So if we can understand what the T cells look like, what the sequence of their receptors are, we can use that to gain insight into these antigens, which will give us insight in turn into disease biology. And we've been very fortunate to be supported by the PLRC, I'm very grateful for their support because it's an innovative kind of collaboration between industry and academia, and organizations like the PLRC are able to move quickly and support some of these types of structures without a lot of the bureaucracy and red tape that you see in some other funding mechanisms. So our goals here are to identify disease-associated immune repertoire features and ultimately leverage those to understand these target antigens and use those to go towards new therapeutic targets and biomarkers. A little bit about T cell biology first before I show you some of the types of data we're generating. So at a high level, you can think of two types of T cells, CD4 and CD8 cells, and this refers to particular molecules and receptors on their cell surface. So CD4 cells are kind of orchestrators of the immune response, and they're often referred to as T helper cells because they secrete these cytokines, which are very powerful immune messengers that control the immune response and activate cells or suppress cells, tell them what to do. There's also CD8 T cells, which are effector cells, kind of the business end of the immune response. And these are cells that can actually directly recognize foreign pathogens. They can recognize virally infected cells and then kill those cells. They produce these effector molecules, including granzymes and perforins, to directly attack those cells and perform what we call immune surveillance. So in a typical immune response, you'll have these types of cells circulating through your body, and they each have a different T cell receptor that's specific for some set of antigens. And if you encounter a foreign antigen, like a pathogen, a virus, a bacteria, that antigen gets presented by what are called professional antigen preventing cells, and T cells that are specific to it become activated. And when they become activated, they expand. So they divide, They create more copies of themselves, these sister clones. And this is what actually works to control an infection. So you get this clonal expansion, many copies of a particular cell with a particular specificity that might attack the infected cells or release effector cytokines to activate the immune response. And once that infection is resolved, immune memory is generated. So these cells differentiate into memory cells that can then activate a secondary response. Important to remember that individual cells with a given T cell receptor and specificity will expand upon activation. Now, the T cell receptor is this molecule that is what actually interacts with presented antigens. So parts that are recognized of foreign peptides or self-antigens, but typically through a process called immune tolerance, autoreactive T cells are weeded out by the body. And you're left with a diverse immune repertoire that can recognize foreign pathogens. The mechanism for generating this diversity is quite remarkable and involves this combinatorial rearrangement of actual gene segments within the T cells. So you can think of these as like little building blocks, which in every individual T cell get recombined into a particular chain. And this sort of process can generate a huge amount of combinatorial diversity, so trillions of possible TCR sequences, with each sequence being related to the structure of that T cell receptor and dictating the specificity of the antigens it recognizes. So you can see here a structural model of a T cell receptor with these two chains, the alpha and the beta chains, with these variable regions that are produced by this rearrangement at the binding interface. And that recognizes peptide antigens presented on the surface of antigen-presenting cells. So it's this sequence and these different TCR sequences, T cell receptor sequences, that dictate antigen specificity. Now, as I said, normally in a typical immune response, you will encounter an antigen and T cells reactive to that antigen will expand. They become effector cells, the antigen gets resolved and controlled, and that generates immune memory. And this happens also when a person is vaccinated. You get this expansion of specific T cells, and you get the generation of immune memory. But in certain settings, including chronic infection or cancer, where there's a persistent antigen, these T cells are being continuously stimulated, and they become functionally exhausted, as Liisa told you about. So they're no longer able to carry out their job of controlling this infection. And you enter this persistent state where the infection is continuing, the T cells are still there, but they're exhausted. And in cancer, there's been a great deal of success in reinvigorating these exhausted T cells with things like immune checkpoint blockers that reactivates an anti-tumor response. The finding of exhausted T cells in ME/CFS and long COVID and the other studies that, especially in long COVID, are showing evidence of persistent virus, suggests that there are persistent antigens that are driving this functional exhaustion. And we would really like to know what those antigens are. And also, there can be autoreactivity caused by activation of T cells that are cross-reactive to self-antigens. So understanding the antigens that the T cells are reactive to can give us a handle and an entry point to look for potential autoreactive T cells as well. So this is very much work in progress. So I'm going to give you just kind of some vignettes and flavors of the types of data that we are generating to give you an idea of what we hope to learn and what we are learning and what we hope to learn with this project. So this is a cell state map. This is called a UMAP. Each dot here is an individual cell. And this is a map of the gene expression data from the single cell analysis. And essentially with this sort of map, similar cells cluster together. What you see here is what are highlighted are T cells with what we call expanded clonotypes. So these are ones they have been activated and expanded, and there are many copies of these cells. This is expression of particular marker genes. Down here at the bottom are particular immune checkpoints and T cell exhaustion markers. And what you can see in this subpopulation of expanded cells, you see markers of activation and exhaustion. This is not something you typically see in the blood of healthy people. If you look here, this is showing expression plots of these expanded clonotypes. So for instance, here, the 18 cells with this particular TCR express these particular genes. And you will see here some notable features. CTLA4 is an immune exhaustion marker expressed here on several of these clonotypes. You don't normally see that. And you see both CD4 and CD8 cells, as well as some of these double positive cells in these most expanded clonotypes. Again, this is unusual, and it's not something you would see a lot of in healthy people. And these cells can have particular properties that might make them more prone to autoreactivity because they have both types of immune receptors and can interact with different types of antigens and might not have gone through the same sort of selection processes that normal T cells have. So we're very interested, and Liisa's lab has been working on these cells for a long time. We're very interested in understanding what these cells look like in ME/CFS and long COVID. In addition, one of the unique features of the cohort that Liisa's lab has assembled is they've been working with some of these patients for quite a long time. You could see the clinical trajectories of some of those patients, and they have archived samples from times when these patients have been more sick or relatively better. So what we are doing is looking at cells from these patients at different time points in different immune compartments. So what this is a clonotype graph of T cells, so T cell receptors. So if there's only one copy of a particular T cell receptor, a particular sequence in this population, so this is several thousand cells, it'll only show up as a single dot. If there's an expanded clonotype, it shows up as one of these families with many members. And what you'll see, so we've been looking at the unsorted, what we call PBMCs, peripheral blood mononuclear cells. We've also been looking at sorted populations, enriched populations of cells from these donors, including CD8 T cells and these CD4, CD8 double positive T cells. And you can see some of these populations are very clonal. This double positive population is highly clonal. So there's some very expanded T cell clonotypes that likely recognize particular antigens and are being driven by the presence of particular antigens in these patients. So we're quite interested in understanding what these expanded clonotypes are recognizing. Also, we can go back in time with archived samples to earlier time points. This, for instance, is a time point when this patient was relatively sicker as compared to this more recent time point. You can see the PBMC population is more clonal here than it was here. So these are the types of studies that we're doing. Here's one finding from this very clonal double positive population where you can see what we call preferential V gene usage. So these are the segments that make up the T cell receptor. Normally there'll be some fairly uniform distribution here. In this case, you see certain V genes are used preferentially, and so it's a skewed repertoire. Our collaborator has done analysis of these TCR sequences, and you see particular motifs. So these are sequences at the binding interface of these T cell receptors that recognize antigen. So, but when we see these kind of enriched motifs and preferential V gene usage, it's very suggestive of an antigen-driven immune response. And so what we're actively doing right now is trying to mine these sequence databases, compare to databases of known antigen specificity, and go from T cell receptor sequence to antigen. And we're also trying to do that experimentally. So to summarize, we see signs of T cell exhaustion and antigen-driven immune responses in ME/CFS and long COVID. We're able to go from single cells to identify functional cells and identify their T cell receptors, and we hope to use that to identify the antigens driving the persistent immune response and understand the immunopathology better. And we're working on a couple of main areas now, definitely expanding the cohort from patients in healthy controls and then both computational and experimental identification of target antigens. These are the folks involved. As I said, this is supported by the PLRC. It's a close collaboration with Liisa's lab and our collaborator Dario Gersi's lab at the University of Nebraska, working on T-cell receptor analysis. And these are the folks at HiFiBiO involved. And I'll just say, it's been very gratifying for me and one of the most, exciting and fun experiences I've had in science, being able to work with this team and in this format with the patient representatives so closely involved, and being able to work in an area that we really think can have translational relevance. And happy to discuss more in questions.
Yeah, it's really exciting work. And I have to say, I was part of the panel, the funding panel too, for PLRC. And I think that the work that you all are doing just shows how important it is to have patients involved, and how, particularly for choosing which projects to fund. So I just had to give them a shout out. So we're going to hear from the patient reps next, Megan and Rivka. We'll start off with Megan.
Yeah, I remember messaging Tess about how excited we were about these proposals. So yeah, it's been really exciting to work as a patient representative on this. Again, just to reintroduce myself, I'm Megan Fitzgerald. I have a doctorate or PhD in neuroscience and neurology, and I've conducted my own research for many years on neuroscience, neuroanatomy of psychiatric disease, and regenerative medicine, stem cell work. But in March of 2020, like so many of us, I developed symptoms of COVID. I had a pretty severe acute course of the disease, and subsequently developed the condition that we all now know is long COVID. I began getting involved with PLRC in August of 2020 and joined formally in December, volunteering on research projects, and just, kind of moved the lens of the focus of my research towards my own disease at that point. And I think that, I'm so excited about Liisa and Roshan's work, because I see this work as being kind of integrative of a lot of the various hypotheses of the causality of long COVID. The immune defense system, it can be a culprit that can induce things like thromboinflammation and blood clotting, and prevent the restoration of fitness after acute COVID and in patients with ME. And patients with long COVID, we show signs of immune dysfunction and autoimmune antibody production. And so I really think that a better understanding of this immune dysregulation is going to lead to a better understanding of the etiology of our disease and symptoms, and hopefully better diagnostics and lead to good treatment targets in our lifetime. And I just wanted to speak into, it's been so great to work specifically with Liisa and Roshan. As they've mentioned, they've been touched by ME in their own lives, and they really get it. It's, this is more than a day job to them. It isn't, you don't get that feeling from all researchers, even if, they're putting in the hours and the time. There really is just an understanding of these conditions and a passion to work on them. So I want to say publicly, thank you all for your hard work, and they're bringing this expertise and years of dedication that they've had to ME research now to those of us, with long COVID, and this is just tremendously valuable. And my role in this project, we've spoke about the importance of having patients involved and conducting the research and informing the research. And I hope I've been able to provide some additional support and insight as, someone who's been living with long COVID. And it's just been tremendously rewarding to me as well, as someone who has a lot of laboratory training and scientific background, to be able to continue to contribute, now that I'm less able to be able to go into the lab, put in the long hours like Anna is putting in. And I'm so appreciative of that, Anna, as well, for you and Nure just doing that work, really holding everyone to like a really high standard of stringency with the scientific data. And it's so fun to come to these meetings with you all and to see this, to see this progress and to be a part of it and to inform it. And I think with that, I'm going to pass this on to Rivka, who is a fantastic patient advocate, many years of experience.
Thank you so much, Megan. First, I want to say thank you to Research Renegade and Remission Biome and the Patient-Led Research Collaborative. So I'm Rivka Solomon. I'm in Massachusetts, and I'm one of the 2 patient reps on the 2 grants that are funded by PLRC. The first is with the Selin lab alone, looking at T-cell dysfunction in ME and long COVID. And the second, as you've heard, is with the HiFiBiO and Selin lab collaboration that Roshan discussed. So I am an ME advocate, and I've been one for 30 long years. And of course, now I'm a long COVID advocate too. And in that capacity, I work with ME and long COVID organizations. I organize events. I work with congressional staffers, the media, and so much more. My personal history is going to sound familiar to many of you here. I got ME in college at age 21. And I got it after infectious mononucleosis from the Epstein-Barr virus. And I am 61 now. So do the math. That means for decades, I've been living with disabling exhaustion, cognitive dysfunction, and post-exertional malaise. In fact, after this organizing of this event and speaking, I'll probably be collapsing, which is the life of an advocate. I'm often homebound and bedridden and often do my advocacy from bed. So I'm going to shift in this talk. We've been hearing a lot about the science, and I'm going to shift away from the science, and I'm going to share how I, an ME patient and advocate who's not a science person at all, became a member on 2 research teams. So it started in 2018 and 2019 with MassME, which is the Massachusetts ME/CFS and FM Association. I helped to organize and lead 18 ME awareness raising and medical education events on college campuses, at hospitals, and other sites. In fact, just to brag, we organized 2 of the biggest in-person ME events ever held. One was with 370 people who came to see the ME film Unrest. And the other event was with 650 people in person who attended a panel at the MGH Institute for Health Professions, focusing on ME. Then in 2019, at the end of one of these 18 events that we organized, this event we held for a Massachusetts state agency that helps people with disabilities. At the end of that event, Drs. Liisa Selin and Anna Gil introduced themselves to me. They told me they were running a 2-woman viral immunology lab at UMass Chan Medical School, and that they were studying ME, what they said, through the back door by studying Epstein-Barr virus. And of course, that was the back door that I got ME. They said that they had some unique findings regarding immune dysfunction in both multiple sclerosis and ME, and we all know that Epstein-Barr virus is implicated in both. And then Liisa told me the kicker. She told me something that really moved me. She said that she'd been living with ME for over 40 years, and wow, right? Well, that was it. Right then and there, I fell in love with Liisa and Anna, and ever since I've been helping them to bring their unique immunological findings on ME and now long COVID to the world. First, I introduced them to ME researchers and clinicians, helped with press releases, media outreach, fundraising, organizing meetings, including meetings with members of Congress. And then in 2021, the Selin Lab was awarded an NIH grant to study T-cell exhaustion in ME, which I believe no one else was doing at that time. And now in 2024, the number of other researchers who are studying T-cell exhaustion in ME and long COVID has increased, including researchers at the NIH. When we learned that PLRC, the Patient-Led Research Collaborative, was offering grants and mandating that patient reps be part of the team, an idea we all loved, The Selin lab generously included me as one of the patient reps, and then the PLRC assigned this lovely patient rep that you just heard from, Megan Fitzgerald, who is clearly such a contribution as a neuroscientist living with even living with long COVID. So before I tell you what it is I do as the PLRC patient rep, I just wanted to share with you a little bit about my relationship with Roshan. Roshan, I have known for 6 years, thanks to our mutual work with MassME, and I already valued him so much because of his past work as a science communicator with the MassME Research Club. But I also deeply respected him because he was a caregiver to his wife with ME. So last year, when Roshan asked me for my thoughts on potential collaborators for a new PLRC grant he had just secured, we were already working on our PLRC grant at the Selin Lab. It was really a no-brainer to suggest that the collaborator be the Selin Lab, because personally, I love to work with Roshan. So I thought, well, what better than to work with him in this capacity? And it soon became clear that it was a match made in heaven with these researchers working together. And for continuity's sake, we kept the same PLRC patient reps, me and Megan, for the collaborative research grant. Now, so what is it that I, a completely non-science person, do as a patient rep? What do I do on these 2 research projects? Well, I continue to help with PR and communications, such as the press release about our collaboration and helping to organize this panel today. Everyone in our team is committed to the ME and long COVID community and the public being informed about the research that we're doing. And we see this panel as one of those opportunities. And of course, I attend, along with Megan, the regular ongoing joint meetings between the 2 labs that take about almost 4 hours a month. I'm the one in the meeting that's always asking, well, can you please explain that in layman's terms? And I also always am sharing what I think the patient community will react to, how they'll feel about something. I'll say something like, don't forget that the patients will want this, or don't, I'm concerned that patients will need that. And I also offer an historic perspective. After all, I've been sick decades, and I've been in the ME community for decades. And because I've been in the ME community for so long, I offer a Rolodex full of ME advocates, ME leaders, researchers, clinicians, and congressional staffers. So before I close, I'm going to say something that sounds similar to what Megan just said. I want to share the 2 things I appreciate about being a patient rep on these 2 specific research projects. The first is that I really deeply respect the people I'm working with. They have integrity, they're brilliant, and they're bringing us cutting-edge new discoveries that are going to concretely help patients. And secondly, I love that most of our team, including the funders, the Patient-Led Research Collaborative, either have ME and long COVID or are caregivers. I mean, it is so wonderful to have the opportunity to work closely with people who are deeply motivated to do this work. And that's the end of my talk. Thank you for listening. Back to Tess.
Thank you, Rivka. And I wanted to thank Megan and Rivka for using your spoons today to come and talk. And I hope you don't crash too hard after this when you mentioned Rivka. So Rochelle is going to be the one who's leading the Q&A, but I wanted to start it off by I've been really curious to see Liisa's slide. So can we start with that?
Sure, not a problem. Happy to do it. Okay, now I got to figure out how to get to the next slide. There it is. Okay. So from the work that we've been doing in this area and my personal knowledge, having had the disease and being a doctor and a scientist, I've been thinking about this, as you can imagine, a long time. And as I see it now, based on our research and that of others, majority of the cases for ME/CFS and long COVID start with an infection, although it is possible a toxin could also be involved. They develop an inappropriate immune response to the pathogen, and this completely dysregulates their immune system, leading to the state of T-cell overactivation and subsequent exhaustion. And at this time, it's unable to clear it's not clear if the exhaustion is being driven by the original pathogen or other persistent pathogens, as I will talk about, or whether there's a component of autoantigen, and it may even possibly vary between patients how much autoantigen is involved and how much persistent pathogens are involved. This could be treated with an immune modulator, and we need to come up with good immune modulators. The second thing that's quite clear to me is that ME/CFS patients have difficulty controlling the persistent pathogens we all have in our body. We've all pretty well been infected with some of the herpes viruses. 95% of people are after 30 have Epstein-Barr virus. 40% of people have cytomegalovirus. Pretty well 100% of people have HHV6. VZV is also probably in the 90% range, as is HSV1. So these are pathogens we carry with us all the time, and they are completely controlled by CD8 T cells. So you need good functional CD8 T cells to keep them under control and lead a healthy life. There are other viruses that may be involved, like enteroviruses. They can persist. JK viruses persist. And even funguses, as you all know, Candida albicans, Borrelia, and now long COVID is thought to persist. There's more and more data coming out to suggest that. This all then leads to the state of CD8 T cell exhaustion. And this is not just a state where the T cells are exhausted, but in the presence of a persistent antigen, like a virus, and the exhaustion, the host keeps trying to keep the virus under control, but unsuccessfully, and it sets up a real immunological mess where you have both inflammatory responses, but you also start developing inhibitory cytokines trying to shut down this immune response. And therefore, you tend to have high type 1 interferon in response to the pathogen. This is inflammatory, but at the same time, you're producing inhibitory or non-inflammatory cytokines, TGF-beta and IL-10. So this is not a good immunological state to be in. These increased cytokines lead to altered metabolism systemically, and that is something infection automatically does, is to alter your metabolism, and that leads to a very high state of oxidative stress. With increased lactic acid and oxygen radicals, and there is a lot of previous research consistent with this. So antioxidants would be useful. The increased cytokines and the oxidative stress directly affect the central nervous system because we know this is a multi-system disease. And that leads to the brain fog, the autonomic instability, and there are some treatments available that work in some of the patients, such as Mestinov for the autonomic instability. Brain fog is still extremely challenging. So when the immune system tries to balance for the overactivation and the T cell exhaustion, it tries to control the persistent pathogens by new ways because you cannot let these pathogens get out of control. And what we're finding is that some patients, in fact, a majority of them will increase this unique population of cells, CD4 positive, CD8 positive T cells that Roshan was referring to. And I think this particular population will be a very useful window for us into trying to identify which pathogens in these patients are driving the problem. And I do think a lot of it is going to be herpes viruses, but it's still good to know in a patient which herpes viruses are involved. But some of them could be enteroviruses, some of them could be other pathogens, atypical mycobacterium, who knows? This all needs to be worked out. But these cells are generally all in an activated state. And therefore, as Roshan showed, they're clonally expanded. So this gives us a way to try to start identifying. Some patients develop cytotoxic CD4 T cells to try to compensate for the CD8. And other patients have increased innate T cells called mate cells. And we've even seen very high frequencies of gamma delta T cells. And we need to sort out how these patients stratify. Is a particular type of compensation more indicative of more severe disease? And is there anything in particular, any pathogen in particular that is more likely to drive the expansions of these populations? The unfortunate thing about the double positive CD4, CD8 T cells, as Roshan mentioned, is that they may enhance the development of autoimmune disease. They'll be escaped. These T cells will possibly escape negative selection in the thymus and may react to autoantigens. So that will be something to keep a close eye on. But at the same time, all of this can also lead to NK and B cell dysregulation. NK cells have been reported to be dysfunctional and non-cytotoxic in ME/CFS patients. And in fact, we have just realized that we can use our biomarker, and even the data that we have already generated, we just need to go back and examine it to look and see what NK cytokine function is like in the various subsets. And we do have a collaboration going on with Mickey Tao and Beth Pollock at MIT, where we're very interested in looking at B-cell immune dysregulation, because their data, preliminary data that we've been working with them on our patient population, shows that most ME/CFS patients and long COVID patients we have lower antibody levels of all subtypes, except they have increased IgE, which would, of course, fit with the mast cell profile, increased mast cell activation and allergy. And the increase of the double, we've also got some data, and we haven't had time to pursue this further, but our initial data showed that these CD4 positive, CD8 positive cells have a Treg-like population within them that makes IL-9 spontaneously, which in healthy people it doesn't. That is an anti-apoptotic cytokine. It helps T cells from dying, so it might be trying to help the CD8s. But at the same time, IL-9 directly activates mast cells. And this is a place where the activated mast cells can be treated with the H1 and H2 blockers. So that, in summary, is how I see ME/CFS and long COVID at this time.
All right, so I guess if we're going to get to questions, thank you so much, Liisa. That was like fascinating, taking notes. But I'm going to start off with the questions that are in the Q&A that have been upvoted the most. There are quite a few questions about Inspiritol. So I'm going to see how we make sure we're not duplicating the same question coming through multiple times. But for all of the participants that are watching, and that I'm going to go ahead and allow you to talk, and then you can ask your question. And then if anybody is not able to answer or ask your question right, then I'll just pause a little bit. And if you don't come on, then I'll go ahead and ask the question for you. So our first person that we'll be bringing up is the most upvoted question so far is from Katie Glass. Katie, are you able to ask your question?
Hi, yes, I'm here. Yeah, so I'm really interested in sex differences in ME/CFS. I'm a member of the Hansen lab, and we're seeing that in almost everything we study. So I'm wondering if you are all seeing sex differences in the CD8 T cell function, the response to treatment, and/or the increased proportions of the different subpopulations of T cells that have been mentioned.
Okay, thank you. That's a very important question, actually. And yes, I agree with you. I think there is a difference first of all, there is a difference in males and females in their immune responses to begin with, even without disease. So one would expect that they might actually respond differently. And there are also metabolic differences between males and females. Most likely they deal with oxidative stress very differently. So it would be logical to assume that you might actually find differences. So I haven't shown the data here, but we have actually taken our patient samples with the CTL profile, a much broader profile of molecules than the ones that I show in this paper, where we look at perforin and granzyme, CD107A and B, and we look at the CD4 and positive, double positive, the CD8 frequency. And we've analyzed them in principal component analysis. And it appears that almost all the ME/CFS long COVID patients are exhausted. They compared to healthy donors. However, they then segregate into two populations. The males seem to have different features than the females in things that we're looking at. And one of the features is that the females do appear to have, and nothing's hard and fast because it's actually the multiple factors that make them segregate in the PCA. But one of the features that they segregate the strongest on is that the females tend to have a higher frequency of the CD4 positive, CD8 positive T cells. And interestingly enough, when we do a correlation with that frequency and severity, we find an inverse correlation. So the higher CD4, CD8 frequency, the lower your overall symptom score. So it seems to have some kind of beneficial role. The males have a tendency to have a greater number of cytotoxic CD4 T cells. And we are finding that that correlates with more severe disease. They also have greater expression of CD107A and B. But all of this, so yes, this is something I think we should all be looking at very carefully, and certainly would be curious to know what differences you're finding.
Okay, thank you very much. Roshan, did you have any comment on that? Have you looked at enough samples to see any sex differences?
I'll say, I think with the single cell analysis, it's still early days, so it's a pretty small number of samples we've looked at. And I think in general, one of the limitations of this study or approach, it's pretty expensive, and so there's going to be a cap on the number of samples, patients, and controls we can look at. This is very much more of a depth rather than breadth study. So one of the things we're working on, and is an active area of effort is as I mentioned, we spent a lot of time trying to build up integrated single cell databases from both the public literature and then our own data, both in cancer and autoimmune disease. And of course, these studies include controls, and so and also applying tools for data integration. So I'm hopeful that for some of these things we can use some integrated control data to see, because certainly like sex differences in age, of course, can have profound impacts on the immune system, to see which findings are most robust and specific to patients. And then, it's because a lot of these things, like, the double positive immune repertoire, it's not like there's a lot out there about, from healthy donors, right? I mean, it's just not known. So I think there's some things that, with the immune exhaustion and all that are already clear that, it's sort of, it's not something you see a lot of in healthy folks, but that's a long way of saying I don't think we have enough data yet to on the single cell level to say anything about sex differences, but I think even, you know, at the end of the day, we're going to have to look to sort of other controls to give us some more confidence and specificity, if that makes sense.
Yes, thank you both so much. Appreciate your time.
Thanks.
Okay, and our next question comes from Julie Sullivan, and she actually has a couple of questions here in the Q&A. So I don't know if you want to ask them both or just one. Go ahead, Julie, anytime you're ready.
Hi, thank you so much. I'm very excited about Inspiritol, and I'm wondering if it's a one-time treatment or if this is a medication that people will expect to take on an ongoing basis. I guess I'm asking how long the results would last.
Okay, so and that's an excellent question. So from the number of patients that have been done so far for ME/CFS and long COVID, we find it's almost like taking, in my mind, it's almost like taking insulin for diabetes. So the dosing regimen is 5 mils 3 times a day. By inhaler, which is a challenge, but when it makes the patient feel better, they definitely are willing to do that. Some patients, perhaps the more milder, but we need a much bigger study. They were able to easily come off Inspiritol after 2 to 3 to 4 months. Their symptoms seem to resolve. However, the very sick patients, both long COVID and ME/CFS, needed ongoing long-term therapy. But the curious thing about that long-term therapy was that they progressed, unlike insulin for diabetes, you stay the same. But with Inspiritol, they gradually, progressively got better and better and better over time as they took the medication. And the longest this study went out to was a couple of years. And there are patients that did take it for a couple of years. And those patients continued to improve in their symptoms in unusual kind of way that they may stay stable for a while and then suddenly another symptom improves. So the answer is not in until we can get actual large clinical trials, but I think it will be some kind of combination of I think the sicker people may be on it much longer than and there is some evidence to suggest that the long COVID patients in this study, they were only, as this was an earlier study, so they were only sick for months, whereas the ME/CFS patients that we had in this study had actually been, the average was 20 years. And changing immune dysregulation and the kind of damage that does to all your organ systems would take a long time. And I also think it needs to be done gently and carefully. And this drug seems to work very well at those levels. So I hope
Can
that
you clarify, just, I think I heard what you said, but double checking that the study went on for a period of time. And so there were some people with long symptoms and severe disease that didn't get better by the end of the study, but stopped taking the drug and then continued to have sometimes stepwise, symptom by symptom, or what have you, improvement. So was that correct, or were they still taking the drug off the study?
For a while, they were taking the drug off the study and they continued to improve. But unfortunately, due to logistical reasons, this is a relatively difficult compound to be made. And it was one small compounding pharmacy making it. And they did not agree to continue making it. So Dr. Salerno is no longer has the option to prescribe it. So people had to go off. So most people who had to go off maintained the level they reached. And the patients who were on, who needed it longer, they continued improving while they were on it. Even when we had to take them off finally. And there are many who would like to go back on it, but it's not available until we can get it through
But it
the
just
FDA.
thinking it's super helpful. In terms of asking the question, it's thinking, so if overall there's some sort of a chronic inflammatory process that's creating a balance towards fibrosis and dysfunction, and then can flip the other way as the immune system is turned down, those continued improvement while off it, where people have been decades without improvement, may suggest it's moving that direction. Just like you could have a chronic wound that you do some things and can get it to start healing, whether it's revascularization or something else, and it can continue forward. It feels like that's a really strong data point.
So right now, what I can say is that people who had many of the people who have been chronically ill, when they went off, they did maintain the improvements when they went off, but they did not, as far as I know, but we haven't really documented that well, they did not continue to improve. And some of them actually regressed, and they felt much better when they were on it. And certainly during the treatment period, people often ask, and this may be one of the questions out there, is how do we know these people wouldn't have gotten better without the treatment over this period, particularly long COVID. ME/CFS, obviously, these people have been sick for 20 years and there's no treatment. Almost all the patients, when they went off, deteriorated. Even if you missed a dose, the patients notice that they would be more fatigued, less functional. They would be lying in bed more. So that's why I say it's kind of like insulin, that it looked to me like it had a half-life of 6 hours. That's why you need to take it three times a day. But these are so early times. I really hope the company can get it through the FDA, get the clinical trials done, so we can really accurately answer all these really important questions. Did that help?
Definitely. And if there's time at the end, I'd love to dive into your mechanism picture a little bit, but let's go through the questions first.
All right.
I had one more question. I'm the program manager of the Brigham and Women's COVID Recovery Center. So we have a large patient base, and they're all, of course, very eager to participate in research. Do you anticipate that people who develop long COVID symptoms from the vaccine will be included in the study? We have a number of vaccine injured folks.
Absolutely. Absolutely.
Wonderful.
We include all patients who have long COVID or ME/CFS, whatever the origin is, whether it's a vaccine, virus, and with ME/CFS, of course, we don't even know the instigating. And it is sad that there are quite a few people who have developed long COVID, which is like ME/CFS, from the vaccine. It's very unfortunate.
Yeah, thank you so much. I appreciate it.
Welcome.
All right, and so our next little bit of questions, it's actually Rivka has several here that she would, it's kind of been sitting on. So Rivka, whenever you are ready.
Oh, thank you so much. So I just want to reiterate before I ask the question that what Liisa said, if there's anyone here from the FDA and you can help, we certainly would love to see Inspirital get approved in clinical trials and everything, or if anyone wants to help with funding clinical trials. My question, which I think others will have here, is about the biomarker test that the Selin lab feels they found, or a potential biomarker test. I guess the question is it a potential biomarker? And your Inspirital paper seemed to show that you were able to use this potential biomarker to track treatment's efficacy.
So, yes, we do feel that this is an excellent candidate for a biomarker for ME/CFS and long COVID. As I mentioned, the PCA analysis that we've done, which I didn't have time to show to you in this, but will be showing hopefully in future talks, and we're working on a manuscript about it, suggests that this would be a useful biomarker for ME/CFS and long COVID, and it is a feasible one clinically, which was important for me as a patient myself and as a physician to try to design something that could be adapted to the clinical setting. We are in the process of trying to find out whether it's patentable and we need to optimize it for clinical strategy with all the appropriate antibodies, but we need money for that. So to get that done and to design a kit that it could be used by physicians all across the country. That's a whole different aspect of this. But yes, I think this plus, as I said, there's a whole bunch of other markers that fill in the whole profile that would be part of that analysis. There is one issue that we still need to do a lot of work with on the biomarker. And no one has asked this question. They asked about sex, but the immune system not only differs between males and females, but it also differs with age. So we need to do a lot of studies to try to, with a larger number cohort, to try to determine the normal ranges, because we are seeing a clear correlation with age. For instance, CD4, CD8 positive cells do increase with age. They do not go to the levels that you see in the ME/CFS, many of the ME/CFS patients and long COVID, but they do gradually, particularly after 70. And I find that very curious because after 70, people also have great difficulty controlling herpes viruses. So I almost suspect that the CD4, CD8 expansion as you age is related to losing control of your herpes viruses. And now you're having your CD8s actually decrease with age. Also, that is I've never actually seen the data the way we have it right now. It is a direct correlation. With age, CD8 is going down, and with age, CD4 positive, CD8 positive cells are going up. And there may be some changes in TNF production with age. It seems to increase with age. So we need to, yes, definitely work out all these things, the male-female sex differences and the age correlates. But yes, we would love to work and are looking for somebody who might want to, if there's anybody interested in trying to help us develop this as a test, that would be great.
I think my other question would be for Anna, who I know always has her nose to the grindstone in the lab, doing the hard work of the lab analysis. So Anna, if I'm sure patients are thinking, what if I get my blood to you? If I can get my blood to you, would you be able to do this T-cell exhaustion test on my blood, and would I be able to see the results?
So yes, at the moment, based on our IRB protocol that is in the process of approval, final approval, but we will accept patients, the ones that are local, meaning in the Massachusetts area, we would say, or can drive, let's say, from Connecticut, surrounding states to Massachusetts. Basically, if they can come to Massachusetts physically and to UMass campus and donate their blood. So yes, we do accept those local patients. But when it comes to the results, at the moment, we cannot share the results with our patients. So there is a possibility that with some other protocols we might, but at the moment, I would say we cannot.
Thank you so much. Let's jump back to Remission Biome for the rest of the questions.
All right, so we'll go back to the Q&A to go to the next question here at the top. There's several Inspirital questions. I'll be kind of sorting through those to see if they've been kind of answered already, because a lot of people are like, where do I sign up for trials? How do I how do I get this stuff? And so there's a lot of those questions, but that's not this next one. I'm just going to be kind of looking through them too to see if we can kind of group them together. But the next question is going to be from Danielle Meadows. Danielle, if you are ready to talk, anytime you are ready.
Thank you so much. Thank you for presenting this work. It's all very exciting to see, and thanks to everyone that's been involved. I think my question essentially builds off of what Rivka was just asking in the biomarker relation. In addition to some of the sex and age differences, have these indicators of T cell exhaustion been explored in similar diseases so that we can understand if the results are unique to ME/CFS and long COVID?
Okay, yes, that's a very important question as well. So they've been compared to healthy controls, and it can be difficult, as you all know, to find healthy controls, particularly when you get to older people. But we have very recently started looking at this biomarker in our other studies in multiple sclerosis. And that, of course, is a related disease where there appears to be a problem with controlling Epstein-Barr virus. And in fact, in that disease, we are finding a level of T cell exhaustion. It does not appear to be as severe as ME/CFS and long COVID, but there does appear to be a level of exhaustion. Unfortunately, you need a lot of money to try to design a study where you start recruiting patients with other diseases, or access to a biorepository. Now, that is another question for us. So all the data I've shown you so far has been done predominantly with fresh blood. We are in the process of doing the studies, and all our data suggests that we can do these same biomarkers on frozen blood. But we're still optimizing the conditions, and that would be particularly important for vaccine trials. And then we could potentially use some if we, with the being able to use it on frozen samples, we might be able to start comparing it to a blood result in other samples of known disease, like rheumatoid arthritis or lupus or some other, if someone had the samples, because thus far, yes, it costs a lot to do that, and we have not had the bandwidth to do that. But that being said, even if this finding were found, for instance, one place I can envision you might find it is cancer patients. Now, cancer patients are the patients who've been described to have T cell exhaustion within the tumor. But I don't know how closely people have actually looked whether they have global exhaustion or is it just in the tumor. So this biomarker test might actually be very useful for many other things. And It does not negate it as a biomarker. Even if other diseases have this finding, it does not negate it as a major immunological abnormality in ME/CFS and long COVID. You just combine it with your clinical symptoms to make the diagnosis.
I'll just add a couple of points to that. Yeah, I think one of the things with the single cell analysis we're hopeful to do is compare to data from other diseases, again, integrated published data or an internal data. I think we need some more numbers on our study to really have a useful integration there, but I think we will be able to get there. And another, I think, really important thing here, and related to what Liisa just said, I think, especially in these diseases where there's so much heterogeneity, it's important to get to molecular definitions of patient subsets, because these are essentially defined clinically, symptomatically, and there's tons of heterogeneity, certainly in cancer and in autoimmune diseases in particular. I think a recent, the Accelerating Medicines Partnership, their recent paper on RA, where they did high resolution single cell profiling, found 6 endotypes, basically different disease states with different responses to treatment in a relatively well studied disease. So I think even if it's operative in a subset of patients, but we have a test to look at it, even if it's not specific, I think that's a major step forward. And in cancer, you really see that exhaustion with, it's antigen specific T cells. They can be circulating, but it's those ones that are the ones being stimulated to the point of exhaustion.
And importantly, it seems like if chronic inflammation is the endpoint of a number of these things, whether it's radiation, chemo, Lyme, viral, what have you, then the idea of a marker such as a CD4, CD8 positive clonal subset of immune cells would make sense as the body's common reaction to an ongoing chronic inflammation, or maybe there's some other driver. It doesn't have to be, the trigger may be distinct, but all those triggers can lead to one place. And it goes back to Dr. Selin's picture around the oxidative stress and change in metabolism. And we see people going to glycolysis from TCA, we see exhaustion, and all of those could bunch into the same common pathway at the end. And then how do you open that back up? So for example, like dimethyl fumarate in MS is a really good NRF2, keeps NRF2 around it from breaking down, and balances more towards build from breakdown, and just from a biochemical standpoint. And so I think we're starting to zero in on that, the common path that could be happening from lots of different triggers.
Yes. Yeah, I agree with that. Yes. I think the research that we're doing is applicable to not only ME/CFS and long COVID. And I think even the treatment in Spiritol might be very useful in many other diseases where oxidative stress is a major problem, because it is so useful there. Unfortunately, we're still quite a ways from there, but I actually am quite amazed at the potential for this drug and giving it the way we do, by nebulizer with small doses, and the fact that it can get into the brain when you give it by the lung. This could even be a treatment for MS, and I know our collaborator Carolina Iannetta is very interested in trying it in MS, but we really need to get it through the FDA and get it to clinical trials so we can pursue what it can do. And so there are so many medical diseases, as you say, that have this common pathway of severe oxidative stress. So this is not the only application for this.
One point I want to add to John's point about chronic inflammation. I think one thing will be interesting as we learn more about these cells is there an innate-like quality that develops where cells are more sensitive to a non-antigen specific stimulation and then the cytokines they produce, are they more general in nature? And that's something I think that's being seen in certain, at least subgroups of some autoimmune diseases, with the changes in the effector molecules that are being produced, that they can be more inflammatory and less targeted in nature.
And if you walk down that pathway, there's a mechanistic path where you can get to a trigger starts the process of inflammation, and then you get nets and microclots, and then that's causing local hypoxia. When you get local hypoxia, you end up driving HIF-1 into the nucleus, and as a result of that, bias the system towards spitting out more cytokines and more ongoing chronic inflammation. And so theoretically, if you could get the blood flowing, and whether it's smooth muscle relaxation or breaking microclots and so on, that could push it the other way. And if we approaching it as that balance between HIF-1 and NRF2, and starting being agnostic of what is the driver by the disease, but rather at the process, you don't really need an ongoing viral or spike antigen to keep that inflammatory loop going.
Yeah, I think we need to better understand the disease state to know the
Just anecdotally, I had bone cancer when I was a kid, and I had some lung radiation from it, and all my life had a PO2 or a pulse ox of 92%, and then ended up with long COVID in 2020, and it's a longer story than time now, but ended up decided to take metformin for it to try to if there was some leg wound stuff going on, to try and work with that. And about 6 weeks into it, brain fog cleared, and my PO2 started going up, and at this point now 2 years later, my pulse ox is about 98, and it just, for decades was lower than that. And so this idea that there's an irritation that creates an ongoing long-term fibrosis is, I think, it's hard to study, but I think it's there.
Mm-hmm. That's remarkable.
All right, so we're gonna go on to some next questions, and I've just got a couple like little things to remind people of. We're going to keep going until everybody's too tired to keep going. So when everybody starts tapping out, we are also recording this. So if you're one of the participants and you are not able to stay on, we know that there's so many of us, we just only have so many spoons. That will be recorded, and it will be up on the Remission Biome YouTube as soon as we have that downloaded and put in there. And then we'll be working on transcripts too for that. And that's a little bit slower process to make sure we have that, but that'll be there as well eventually. But then so we do have it recorded. So if you do need to drop off, that's perfectly fine. It's not like this is the last chance you'll ever get to get this information. So do remember that. Also wanted to mention that when Liisa talks about exhaustion, she's referring to T cell exhaustion, not necessarily just being like PEM or otherwise exhausted. She's meaning it in a science sense of T cell exhaustion. So just kind of keeping that in mind when you're hearing it talk. She'll sometimes, if she means physical exhaustion, she'll say that specifically. So so I just wanted to make sure on that. Also, there's several questions about Inspiritol that I'll kind of like group together just to get this idea about, do you have any idea on a timeline on when the FDA may approve it, when trials might start, and then also how might people be able to follow that if there's any information on where they might be able to find information on that as it is being updated?
Okay. So just to make clear to everybody, I don't work for Inspiritol. I'm a non-paid consultant for them, so I don't always know exactly everything that Inspiritol is doing, but they are actively pursuing investors so that the in order to have the clinical trials, they need the investors so that they can start mass producing this drug, because it really cannot be easily produced in a compounding pharmacy at the level you need for clinical trials. So they are still actively pursuing that, and hopefully they will have good results soon. They are actively still pursuing their FDA application and in discussions with the FDA, but there's still no, the FDA, so, or if you don't know the FDA, you kind of never know what the FDA is going to do, especially when you're a tiny company as they are. And the main concern of the FDA, the curious thing about this Inspiritol is that the 5 compounds in it you can buy at your local vitamin shop, but not in the nebulized form. Of course, you need the highly purified forms, but you can still buy those as well. The FDA is mostly, I think, concerned because it's given by nebulization, given into the lung. So they want to be very careful with that, and all of that needs to be worked out. In Australia, apparently, all 5 compounds are singly approved to be given by nebulizer. And the company is working on possibly doing a clinical trial there because it's a shorter trip with the FDA just to combine the 5 into one when they all are, but in a nebulizer. So they're still working on these, but there is no definitive, unfortunately, at this point, there's still no definitive timeline, and anything anybody can do to help with that would be appreciated, either with helping them get it through the FDA, or if there are any large investors out there that, because this is a very small startup company that is pushing this forward. So unfortunately, I don't have a clear answer on that, but they are making progress. And to keep up to date, I guess certainly as soon as there's a clinical trial, we're in the process of setting up a website for my lab. It's not done yet. We would certainly notify people if there was one. And I'm sure the Inspiritol, their website would notify if there was one. Dr. Nancy Klimas is very much a part of our clinical research, and I'm sure her websites would notify you of that. We have submitted a research proposal to the NIH for a clinical trial, but at this point it's been put on the back burner, has not been funded. So they're still working on avenues, and we're working on avenues to try to get it, but we're not there yet. So that's as far as I can answer at this moment.
Basically, we want to have it faster, but can't promise anything. And so people, if they would, Inspiritol, do you think they would have any publication or like a source you can be like on a mailing list for them to say like, we're doing it now, and people can find out, or just kind of just try to follow publications that we can see?
Inspiritol got funded and we were doing it now, they would find a way to let you know. And I don't know if people want to write their representative or whatever to try and get NIH and the FDA to move on this. It would be great. Or if there's any, so I don't understand. Rivka understands these processes, but so much of this is, the paperwork behind getting FDA approval and getting clinical trials up, understandably. is complex, but I really hope that we can get it out there sooner, because I really think it would have actually saved lives with acute COVID, because it is also very effective against acute COVID. And many people, if they were able to get on this with acute COVID, might not even get long COVID. So I really hope that we can just keep pushing and get this done, but I don't have a clear answer at this time.
I think that there's no harm in people emailing. So we all know that the NIH says they're going to do clinical trials, so there's no harm in emailing the head of NIH or the head of NIAID or the head of NINS. That's the National Institute for Neurological Diseases and Strokes, and the National Institute for Infectious Disease. Sorry, and Allergies and Infectious Disease, and telling them that you would like to see Inspirital trialed as one of the trials.
All right, and there's like another question on here, but the person who had asked it has already had to drop off for the day, so I'll go ahead and ask it. And this one is for Roshan. For healthy controls, are you using COVID naive controls or recovered COVID controls, or both? And what sample sizes do you have for the different groups?
So these are all coming from Liisa and Anna from Liisa's lab. So as I said, we're still in progress. I think what we're working towards is going to be on the order of 10 to 15 per cohort, so pretty small. But as I said, that's also going into multiple, not cell types, but enriched cell populations per donor. So it adds up. And as far as I think there will be within that, unless they're frozen from prior, I think they're not certainly necessarily going to be COVID naive. So there's some, as I said, data comparisons we can do to publish data from before, and we have some internal samples from the before times to look at. But I think definitely prior COVID exposure and certainly COVID vaccination are going to be complicating factors. But my view on that is, especially for SARS-CoV-2 specific cells, which at this point most people have, it's the functional state that we will be able to look at that can help us sort of associate them to symptoms, really, because with the healthy control enrollment, of course, it's based on symptoms, right? So I don't think it's a case where we're not going to see any anti-SARS-CoV-2 responses, in the controls. I think it's a matter of the functional state there. I don't know if that answered
it. For the most part, yeah, I think you got it. I mean, unless Lisa's got something to add.
Yeah, so if I can add something to that. So that is a very difficult question in many ways. Now that almost everybody, the vast majority of people are post-COVID. So unless we go to biorepositories prior to the COVID pandemic, it's hard to get and including the vaccine. And one of the things we try with our healthy controls very hard is to do them at least 3 months or more post any viral infection. We don't want them to be in the midst of a viral infection of any kind, including SARS-CoV-2. We really try to get them healthy. But for any new enrollees, we do take their history of COVID, and we have a record and of their vaccines. But I think some of our earlier data, including in this paper, actually, the majority of the healthy controls or half of them at least, were pre-COVID, because we started this before COVID. But I think now it'll be hard to get non-exposed either to vaccine or virus.
And so another question, kind of trying to get to the questions where people are still here so that I can get to them to be able to ask their question themselves, because several people have been dropping off just because we've been here a little while. So the next one, Cynthia Johnson has a couple of questions. So Cynthia, if you are ready to go ahead and ask your question.
Thank you. This has been very interesting. I missed the very beginning, but thank you for your good work. Yeah, I was curious. I had do you want me to ask all my questions at once since we're towards
Yeah, because I think you've got like
they're
3
kind
in
of
here, right?
interrelated. Yeah.
Yeah, they're all kind of
and some have
interrelated.
been slightly addressed. If you've been working in collaboration with private groups, for example, or will be with like the Ludwig Cancer Research on T-cell exhaustion, comparing ME/CFS and cancer, I think you touched on that a little bit. And then many people with ME/CFS, myself included, are cancer survivors. And I wondered if somehow this could be opened up in terms of treatment opportunities via oncology and other specialties, sometimes when we're waiting for FDA approval on drugs like this. And then my last question was, I too believe that we need to look at people over 70 or 70 and older. And we just happen to have an abundance of patients right now, sadly, I think, due to the neglect of ME/CFS that are, 65 and over, 75 and over, and aren't being included in research. So I was wondering if you thought how we could start capturing that cohort, not only for your research, but in other ways. So those are my questions, if anyone wants to.
Okay. So the first question about cancer, we'd certainly be happy to do collaborative work with any major cancer foundations or groups. I do think it would be, as I mentioned earlier, that it would be important to look at this biomarker and the approach we're taking in cancer patients to, one, even to see if they have a defect, because I actually believe one of the reasons there is a higher frequency of cancer in ME/CFS is due to this T-cell exhaustion. Because not only are the CD8 T cells important for controlling persistent viruses, they are also very important, along with the NK cells, for surveilling your body for cancerous cells. So if they're exhausted, you probably are at a greater risk for cancer. So that is definitely, and there is even a possibility that enspiritol might have a usefulness someday in cancer if those investigators are interested in looking at that. And if combining with that type of group helps the company to get it through the FDA and funded, that would be great. And I'm sure they're here listening. And we'll think about this, and we've certainly thought about it before. And the second question was studying people older than 70. So we definitely have studied a person older than 70 because we've studied me, and I'm now 72. But we also study other people over 70. But it is much more challenging to study people over 70 because the immune system does change a great deal at that age. And that being said, this might also be, I don't want to keep selling my biomarker, I'm sorry. it might be a useful test to see how well, if we can get this done and get it out there, to see how well a cancer patient's or a healthy donor's immune system is functioning at any time, especially if they're dealing with cancer. It might be a diagnostic criterion of what you can decide what treatment to use or how serious they are. It's I do think it may be there, but it needs to be done. So I agree with everything you said, and we will try to look. We do try to look at people of all ages.
Thank you so much. This has been really informative and hopeful, actually.
I'll add, we do a lot of work with cancer because, several of our programs are aimed at that. And then there are certainly links with, autoimmune disease like Sjogren's syndrome, high risk for B-cell lymphoma. About 25% of severe Sjogren's syndrome patients develop lymphomas. And there's this dysregulation of the B cells and then this chronic stimulation, and they can go on to become cancerous. I think if part of the question was aimed at, cancer treatments for ME/CFS and long COVID, I think I agree with what Liisa said before, on checkpoint inhibitors and things. I think we really need to understand the mechanism here. One of the things, as a caregiver, going through the journey through the medical system with my wife, that was kind of surreal and frustrating was, doctors, wanting to try something would try sometimes diametrically opposed treatments, some anti-infective, some antiviral, and some immunosuppressant. And in the case of cancer, with immunotherapies, you know what you want to do generally. You want to stimulate tumor-reactive T cells, and that's what those treatments are oriented towards. I think we want to move from kind of phenomenology with T cell dysfunction and exhaustion to an understanding of mechanism, and how it's involved in disease pathology, and that can help guide what you want to do for treatment.
Thank you.
All right, and next question, and I think this person has also had to leave already, so I'll go ahead and ask her question. And this is, I think this would be more for Roshan, I think. What is the possible role of IVIG for treatment, and which patients with ME or long COVID may benefit?
Yeah, I know, again, from my wife's experience and her doctors and urologists, we're saying that they, see about 30% of IVIG patients respond to treatment. Again, she tried it. She had a very, very bad reaction. I mean, that was one of, kind of the inflection points with her illness. And I know different people react differently. I guess we haven't talked so much about the B cell side of it, but I think, as I understand it, part of the, mechanism of IVIG, although it's still not clear, is that there's some feedback, dampening down of the antibody response, the B cell humoral response, and that's another aspect of it that we're looking at. I mean, I would think, trying to understand that and classify patients into ones where you have more of an active antibody response could lead them to be treatments for IVIG. There are other treatments now too, like FcRn inhibitors that are being used in a lot of autoantibody-mediated diseases that inhibit, that promote antibody clearance. So I could imagine some sort of stratification like that.
All right. Okay, looking through kind of questions popping up, and we haven't heard from Anna very much, so we've got a question for Anna that's come through. So Anna, what is it like working on ME/CFS and long COVID with so many people with ME and long COVID?
So It's a very challenging, we are on a very challenging mission, I would say, but we are on a mission. So we are very much interested in helping those people. And I know that we and others, actually, we started seriously thinking about research and finding the cause and mechanism and so on. So we are going into the really good direction. We are just not there yet. So this is kind of like disappointing, but at the same time, when we see those people and how much they suffer, how much for so long, it's really, I would say, to me, it's a huge motivation to help them and try to find either the diagnostic tool or again on the basic science level mechanism causes of those diseases. It will be probably a little bit easier maybe with long COVID, but again, those are two very challenging conditions. So yeah, we are working really hard and trying very hard to help them.
All right, thank you so much. I also want to check in with our panel too, and see how everybody is feeling on, we're hit the 2-hour mark. So we have been here a while. You guys have shared so much. Is there, not sure how you're all feeling about continuing on, or if you want to have the remaining questions, we can send them to you and then you guys can let us know and we can, any answers, and then we can send that to the attendees so that way they can see their questions answered. Or what would you like to do next? And then Tess may actually also comment on that as well.
Sure, I'm open and I'm happy to take questions offline if people have questions afterwards too.
I'm thinking that 2 hours might be a good place to stop. It's been just remarkable. But a lot of us are patients, so.
Yeah, I think that sounds good.
And I'm also willing to answer questions off if someone sends me the questions. I'm happy to try to answer them.
Actually, Liisa and Anna, do you want to put in, and Roshan, do you want to put in your email addresses
We can do
into
that.
the chat, or maybe Tess can send them to people with a follow-up email?
Yeah, we'll send resources in a follow-up email, including the recording and some of the links that were posted in the chat throughout this, and we can share the contact info.
Great, thank you.
To register for our studies, you would need to contact either Anna or I if you'd like to join our study by email. That's our best way of contacting us. And I guess you're going to share that.
Yeah, we can email that.
I also want to say we got through at least half of the questions, so that was good. A lot of times we had a lot of questions on here and there's just so many, but we got through at least half of them, so that's great. And some of them were like overlapping, so it kind of helped on that. So that's very optimistic. We got through a lot. So I don't know, I'll let Tess make a determination if that's where we're at the end here. And then Rivka did just put the emails in the chat too. For anybody who's looking in the chat, you can also look at those there, but we'll also send those in the follow-up email after the webinar.
Yeah, I think this is a great place to end. And I just have to say, I was even more excited about these presentations than I expected. I think you all are doing amazing work. And let's get some more funding to continue this.
Yeah, that is crucial. More funding, more investment in getting our biomarker done and Inspirital into clinical trials. That's very important.
It's so great. I just want to say in closing how it's so great to work with patient-led organizations like Renegade Research and Remission Biome, PLRC. You guys are doing a fantastic job. Keep it up. And thank you for giving us this excellent opportunity.
Thank you.
Thank you so much.
This was fun.
Yeah. Great.
All right.
Really appreciate it.
All right, bye everyone.
Take care, everybody. Bye.
Thank you. Bye.